Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper. These are part B questions. The exam was conducted in the year July 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CSIR Net Life Science being discussed, I have already done that in my channel. You can go to my channel Omni Bioentrance. Search for CSIR in the playlist. There you will find all the question papers being answered. Coming to the first question. Which of the following activities is associated with mitochondria associated endoplasmic reticulum membranes, MAM? So MAM, they are subcellular compartments that connect mitochondria and the endoplasmic reticulum. The endoplasmic reticulum is connected to the outer membrane of mitochondria, and they play very important roles like in calcium homeostasis, in regulating ATP production, in lipid metabolism like phospholipid and cholesterol metabolism, in cellular autophagy, in apoptosis, inflammation. So here out of these options, it is not ATP synthesis, it is regulation of ATP synthesis. So phospholipid metabolism is the right answer. Answer is option 3. The next one. Which of the following statement correctly explains the function of a GTPase activating protein, GAP, in the regulation of heterotrimeric G proteins in plants? So whether it is in plants or animals, heterotrimeric G proteins, they get activated upon binding with GTP. If you see here, this is the GPCR. This is G alpha, which is bound to gamma and beta subunits. G alpha is attached to GDP. When it gets hydrolyzed to GTP, the beta gamma subunit separate from the G alpha subunit, and then it processes various signals, downstream signals. So here, which enzymes are the ones that convert GDP to GTP bound form? So it is GEF, or guanine nucleotide exchange factors. They are proteins that catalyze release of GDP and allow GTP to bind. Then there are the other enzyme which catalyze the conversion of the active GTP form to the inactive GDP bound form, and these are called as GTPases. So here what they have asked, which of the following statement correctly explain the function of GTPases, that is GAP, that is they convert the active form, that is the GTP bound form, to the inactive form, that is GDP bound form. So GTPases, they activate G beta protein. That's not correct. G beta is not bound to any GTP or GDP. It inactivates G alpha protein. That's correct, because G alpha gets active only when it is bound to GTP. And what does GTPase do? They convert the active form to the inactive form. So 2 is correct. It directly inhibits ligand binding to GPCR. No, it leads to dissociation of G alpha from beta gamma. That happens only when G alpha is bound to GTP. So 4 is also not correct. Then next one. Which of the following pairs correctly matches the enzyme with its allosteric activator? Phosphofructokinase citrate. Phosphofructokinase, it is involved in glycolysis and its activator is fructose 2,6-biphosphate and AMP. Whereas citrate is an inhibitor, as well as ATP is also an inhibitor of phosphofructokinase. So it is not an allosteric activator, but it is an allosteric inhibitor. So 1 cannot be the answer. Then comes pyruvate dehydrogenase and NADH. Pyruvate dehydrogenase, it converts pyruvate to acetyl-CoA, and here NAD plus is getting converted to NADH. So NADH cannot be the activator, it can only be the inhibitor of pyruvate dehydrogenase. So that is also not correct. Then comes pyruvate carboxylase. So pyruvate carboxylase, acetyl-CoA is an allosteric activator of pyruvate carboxylase and not ADP. Then pyruvate kinase. Pyruvate kinase convert phosphoenol pyruvate to pyruvate, and fructose 1,6-biphosphate, it is an allosteric activator of pyruvate kinase. So the answer here is option 4. Then next one. Which of the following transmitters is synthesized in synaptic vesicles instead of being transported to the vesicle after its synthesis in the cytoplasm? So there are neurotransmitters that are synthesized in the synaptic vesicle. They are not transported to the vesicle from the cytoplasm after synthesis, but they are synthesized in the synaptic vesicles, and one such is norepinephrine. So this is the next points I will be telling only for your information. So norepinephrine, it is released in time of stress, that is flight and fight response. Then norepinephrine, it is part of the sympathetic nervous system. So how is norepinephrine made? It is made from tyrosine. Tyrosine is converted to DOPA by tyrosine hydroxylase. Then DOPA by the action of decarboxylase produces dopamine. Dopamine by hydroxylase converts dopamine to norepinephrine. And norepinephrine can then be released to the presynaptic terminal to the synaptic cleft, or it can get converted to epinephrine by the action of phenylethanolamine N-methyl transferase. So 1 is the answer. Then how many amino acids are present in calcitonin, a calcium lowering hormone synthesized from C cells to the human thyroid gland? It is 32. Answer is option 2. Then who experimentally demonstrated mutations occur randomly? Alfred Hershey and Martha Chase. They were involved in transformation principle, that is DNA as the genetic material. Then Michelson and Stahl, again in DNA replication is semi-conservative. Then Salvador Luria and Max Delberg, they said mutations occur randomly. So it is 3 is the answer. Jacob and Monod with the operon concept. So if you know the other 3, you can happily go for, 1, 2 and 4 options. If you know what they have studied, then you can happily go for the third option. Then next one. Which one of the following statements regarding plasmodesmata in plant cell is incorrect? So they are specialized cell-to-cell junctions. Of course, that's correct. They are open channels that connect the cytosol of adjacent cells. That also happens with plasmodesmata, so that's also correct. The plasma membranes of the adjacent cell extend continuously through each plasmodesmata. That's also correct. Plasmodesmata are extensions of chloroplast. That is incorrect. They are not extensions of chloroplast. They are cell to cell, connecting one cell to the next cell, not with chloroplast. So they have asked for the incorrect statement. So your answer is 4. Then next one. Which of the following statements of the lac operon in E. coli is incorrect? The lac operon is controlled by both the lac repressor and the activator protein CAP. That's correct. Lac repressor is also controlling as well as the protein CAP, catabolite regulation. In there you will find CAP. When glucose is present, when glucose is absent, in all these conditions you will find CAP. Then next one. The lac operon is highly expressed only when both glucose and lactose are absent. If lactose is absent, then how will lac operon be highly expressed? If glucose is absent, lactose is present, then lac operon will be expressed. So 2 is incorrect, and they have asked for the incorrect statement. So 2 is the answer. Third one. The lac operon is highly expressed only when lactose is present and glucose is absent. That's what I told you just now. So 3 is correct. Then in the presence of lactose, the repressor cannot bind to the operator. Yes, repressor will be bound by the lactose. So it cannot bind to the operator. So 4 is also correct. Then in rate zonal centrifugation, a mixture is spun just long enough to separate molecules that differ in mass but may be similar in shape and density. Which one of the following options represents the component that is typically used to create a gradient in this technique? So you want to create a gradient, and the gradient should be in such a way that the molecules will get separated based on their mass. Okay, so this is to create what is called as density gradient, similar to that. So which chemical do you use? You will use sucrose. In cesium chloride density gradient centrifugation, you use cesium chloride to create a gradient of the variation in density or a gradient of density. Okay, so similar to that, in rate zonal centrifugation, you use sucrose. Then in eukaryotes, nucleosome remodelers are. So nucleosome remodelers, they are enzymes that use ATP to reposition the nucleosomes, or their function is to remove nucleosome but not to degrade them. Then they also function in rearrangement of histones also, but not in the modification of histone. Okay. Then methylate histone H3. So methylation is a histone modification. I already told you they are not involved in modification of histone. Methylation of histone is performed by methyl transferase enzymes. So they are not nucleosome remodelers. Acetylate histone H3 and H4. Yes. That is also a type of modification of histone. So nucleosome remodelers are not involved there. So histone acetyl transferases are the enzymes that carry out this process. Then create DNase 1 hypersensitive site. Okay, that's correct. Because here the regulatory protein get access to these sites which are created by DNase 1 hypersensitive site. These sites, the regulatory proteins get access and they can facilitate the process of transcription. Degrade histone subunits. That is not correct. Nucleosome remodelers do not degrade the histone. They reposition the histone. They, or I can say they make arrangements or rearrangements of the histone, not degrade them. Then next one. This was really very simple question. A DNA molecule is completely transcribed into messenger RNA by an RNA polymerase. The base composition of the DNA template strand is given. The base composition of the newly synthesized RNA molecule. So RNA molecule is synthesized from DNA. So you all know the RNA molecule will be complementary to the DNA template strand. And instead of T, uracil will be added in the RNA. So in all the options, uracil is there. Now you have to see only for the base composition. Now G here in the DNA template, it is complementary to C. So RNA will have C there wherever G comes. So what is the base composition of G in DNA template? It is 24.1%. So what will be the base composition of C in the RNA template? It will be 24.1. So where C 24.1 is seen in options 2 and 3. Okay, the other two, 1 and 4, we will not take into consideration. We will see 2 and 3. Now C is 18.5%. That means G will be 18.5% in RNA. So G is 18.5% in 3. So 3 should be your answer. Okay, we will see A also. A is 24.6% in DNA. So U will be 24.6% in RNA. So U. Then T is 32.8% in DNA. So A will be 32.8%. So answer here is option 3. Then next one. Select the correct combination of terms from plant breeding system that represent selfing or promote selfing. Autogamy, allogamy. Allogamy is cross-pollination. So wherever allogamy comes, it is not self-pollination. So 1 is wrong and 3 is also not correct. Then cleistogamy and genesiogamy. Cleistogamy is pollination inside a closed flower, as is seen in the case of groundnut. So 2 is a type of self-pollination. Genesiogamy is also self-pollination. So 2 is your answer. Then next one. Given below are Ramachandran plots for 4 different proteins. Choose the correct pair of proteins, both which are predominantly alpha helical in nature. So as per the Ramachandran plot, you all remember that helix occur in this quadrant. If you see the C diagram, this quadrant is also having helix. This quadrant is also having helix. Whereas in option A, if you see, that is what I told you now is what is there in normal Ramachandran plot. This quadrant will be having beta pleats. Okay. So coming to option A. Option A is having maximum over here. So it should be beta strands. So the major dotted portion, it is on the upper left quadrant, which is for beta sheets or anti-parallel or parallel beta sheets. So A is not correct. Again B, if you see, it is mainly concentrated, the dots are concentrated here. So that must be again for the beta sheets, anti-parallel and parallel beta sheets. But C, if you see, the dots are concentrated here. That is on this quadrant. And that quadrant I told you it is for helix. So C must be one answer. So if you see the options, you can see option 4 is the answer. So D is also it is concentrated over here. So that must be alpha helix. So C and D must be the answer. Then next one, this was your +2 level question. Which one of the following statements regarding structure of water molecule is correct? So the angle between HOH in water molecule is 104.5 degrees. You all know. And the hybridization is sp3 hybridization. So the answer here is option 3. The oxygen atom in water is sp3 hybridization with an HOH angle of 104.5 degrees. Then next one. In the representation of the dipeptide shown below, the superscript -1, that is this one over here, denotes the atom of the previous amino acid, while +1, this one, denotes the atom of the next amino acid. Okay, this is your where your peptide bond is there, CONH bond. The atomic coordinates of how many and which of the following atoms are required to uniquely define the torsion angles psi and phi of the Ramachandran plot. Now totally how many atoms? 1, 2, 3, 4. 4 atoms are involved. Now you will see for the phi and psi. Phi is between NC alpha and psi is for C alpha C. So if you see 4 atoms will be taken into consideration, whereas 2 and 3 we will not. Okay, 2 and 4 we will not see. Now see for the phi. Phi is between NC alpha. NC alpha C-1. Yes, this also you have to take into consideration. C-1 N C alpha will be psi. C alpha C will be psi. So first option is the right answer, whereas in third option it is H over here. So that is not correct. And N C alpha C N plus 1 is your psi. So first option is the right answer. Okay, while explaining, I made a mistake over here regarding the position of the peptide bond. It is over here. Okay, CONH bond. It is here. CONH. I told you it is over here, but it is not correct. I am extremely sorry for that. Just now I remembered that one. Okay, next one. Which of the following statements regarding principles of linkage mapping in plants is correct? Genetic markers would always show higher recombination frequencies when they are closer to each other than when they are far apart. That is highly incorrect. Not when they are closer to each other, but when they are far apart, they show high recombination frequency. So first option is not correct. They have asked which is the correct statement. The genetic distance between two markers is a true representation of the physical distance between them. That is also incorrect. Genetic distance is measured in centimorgan and physical distance is by base pairs. And the base pairs are not going to be equal to your centimorgans at all in any case. One centimorgan corresponds to different base pairs across species. So second is also not correct. An ideal mapping population for a self-pollinating species is generated using polymorphic parents that are inbred lines. Yes, inbred lines will naturally give rise to self-pollination. So 3 is correct. Then an F2 mapping population would segregate in a 1:2:1 ratio for a dominant marker. No, it is 3:1 ratio that it segregates, not 1:2:1. So the answer here is option 3. Then the correct order of the following proteins are recruited during DNA double strand break repair in prokaryotes. So this is just a theory question. If you learnt DNA double strand break repair in prokaryotes, you can happily answer this one. So you all know the sequence. It is Rec BCD first, then single strand binding proteins, then Rec A, then DNA polymerase 3 and finally DNA ligase. So the answer here is option 4. Then next one. Which of the following does not occur during ribosome associated quality control of damaged mRNA? So what is this ribosome associated quality control of damaged mRNA? What is the damage that is happening in mRNA? So this happens in mRNA that do not have a stop codon. That is the damaged mRNA. Okay. So in such situation, what happens is the ribosome will get stalled over there. The nascent proteins that has been translated, they will be marked for degradation. mRNA also will be degraded. And the ribosome, what will happen to that is, it will be disengaged from the defective mRNA. It will not be degraded, it will be disengaged. Okay, that is, it will be released from the mRNA and can be used for future translation events. Now see the options. So they have asked which of the following does not occur. mRNA degradation, it occurs. Nascent protein degradation, yes, that also occurs. Disengagement of ribosome from mRNA, that also occurs. Ribosome mRNA monosome degradation, that doesn't occur. What they mean here is ribosome mRNA complex get degraded. I already told you ribosome doesn't get degraded, mRNA gets degraded. So fourth option is not correct. So your answer is 4. Then next one. In eukaryotic cells, DNA replication is restricted to the S phase of the cell cycle because DNA polymerase is present only in the S phase? No. Origin replication complex recognizes origin only in the S phase? No. MCM helicases gets activated in the S phase of the cell cycle. That is correct. MCM helicase get activated in the G1 phase? No. They are inactive during the G1 phase of the cell cycle and they get activated during the S phase. So 3 is the answer. Then next one. Retinal rod cell cGMP phosphodiesterase is an enzyme with subunit structure as. So this one is having a subunit structure which is alpha, beta, gamma 2. Where alpha and beta are the catalytic subunits and gamma 2 is the, or 2 gammas are the regulatory subunits. The catalytic subunits, they are responsible for cGMP hydrolysis activity and gamma subunit act as inhibitor of the enzyme activity. Now next one. A researcher overexpresses the full-length genomic sequence of rice GAPDH gene under a CAMV35S promoter in transgenic rice. Which one of the following options can be used to confirm transgenic lines using PCR? So what here they have done is, this researcher, he want to overexpress the full-length genomic sequence of rice GAPDH. So what he has done is, he has created a construct where GAPDH is under the influence of the CAMV35S promoter and he has transformed the rice with that. Now he want to check whether he got a transgenic rice. That means whether the rice which he got after transformation is having GAPDH sequence as well as CAMV35S promoter sequence. So first one, using exon specific primers of the GAPDH gene. That will not give the right answer because in the option CAMV35 promoter sequence, what about that? Will that be expressed or not? Whether it is there in the transgenic rice. Only if it is under the CAMV35S promoter, the GAPDH gene will be overexpressed. Now since it is a case of overexpression, I can tell you one more point that these using exon specific primers of the GAPDH gene will result in the internal GAPDH gene also getting amplified. Not only your, okay, you understood. I think you all understood. Not only the GAPDH gene in the transgenic rice will get, which you have introduced, will get amplified, but the internal GAPDH gene already present in the rice will get amplified because it is a case of overexpression. Because already the gene is present in the rice, you are expressing it more. Then using intron specific primers, that is also not correct. Amplification of the promoter region of the GAPDH gene, that also is not going to show you whether it is transgenic or not. Using CAMV35S promoter specific forward and GAPDH specific reverse primer, that will give you the correct transgenic plant analysis. So answer is option 4. Then next one, this was really very easy question. So cell lysate in 1% TX100 was purified over an affinity column. Okay. For what? To isolate a complex with a certain enzymic activity. Okay. The purified enzyme complex was separated on 10 to 50% continuous sucrose gradient. Okay, after purification, that is by after affinity column, you again purified the enzyme. Shown below are the UV spectra using an absorbance filter at 280 nanometers or 260 nanometers. So these are the absorption spectra. So this is at 280 nanometers and this is at 260 nanometers. So you all know that at 280 nanometers what is going to be shown by the UV absorption spectrophotometry is your protein content, and at 260 nanometers it is either your DNA or your RNA content, or I can say in general nucleic acid content will be shown at 260 nanometers. Now which one of the following combination of molecules would generate the spectra shown above? So this will give protein and this will give nucleic acid. So out of these options, both are protein. First one not correct. Second one protein and RNA. Yes, that's correct. This is protein. Second one is RNA. Protein, lipid, protein, cholesterol. They are not correct. Then next one. Brassica juncea has bisexual flowers. Okay. A mutation in the mitochondria lead to cytoplasmic male sterility. Okay, what is cytoplasmic male sterility? It is inherited maternally and it prevents the plant from producing functional pollen. That is cytoplasmic male sterility. CMS can be restored by a restorer of fertility gene RF, which is a nuclear gene. Okay, now this cytoplasmic male sterile plant, if you want to restore them back to normal fertile plants, there should be a gene called as the restorer of fertility gene RF, and this is coded by the nucleus and they can restore the male fertility back in a CMS plant. Fertility restoration is a dominant phenotype. Yes. Okay. Now what they have done? A CMS line is crossed to a homozygous RF line. CMS line, I told you, they have told you it is a mutation. So it is going to be recessive. So out of this photograph, this one is the CMS line and this one is the restorer of fertility line. The obtained F1 progeny, this is the F1 progeny, is self-pollinated. That is, you get your F2 progeny. F1 upon selfing, you get F2. So this is the selfing process. These are the F2 individual progeny that you get. What percentage of F2 progeny will be male sterile? So which will be CMS. Now if you cross these F1 progeny, you will get RF, 1 minute. RF, which is not CMS because it is capital RF. Then here if you see again capital RF, small rf, not CMS. Again capital RF, small rf, not CMS. But this one, this is RF, that is a CMS line. And they have asked what will percentage of the progeny will be male sterile. So 1 out of 4 will be male sterile, that is 25%. So your answer comes to option 2, that is 25%. Now next one. Which of the following option has the correct set of components for assaying reverse transcriptase activity? Now reverse transcriptase, it uses RNA template and synthesizes DNA. Okay, so what has to be the template? RNA template. Now you can see in option 1 and 2 it is DNA template. So option 1 and 2 are not correct. Here you can see DNA template. So they are not correct. Okay, 3 and 4 we will see. So since you are synthesizing DNA, you should have DNA primer. So DNA primer is there. Here they have mentioned RNA primer, so that's not correct. So 3 is the right answer. So DNA primer, then DNTPs. DNTPs have to be supplied because it is DNA synthesis. Then buffer, then RNA template. So 3 is the right answer. Now last one. A student counts the number of seeds produced by 10 different haploid Arabidopsis plant and obtains the following data. So when you see the data, it ranges from 0 to 3000. That's very wide range data. Okay. So and mean Which of the following is the best measure of central tendency for summarizing the above data? So mean if you take into consideration, it is influenced by very large numbers. Okay, so mean will not give you a right measure of central tendency. Whereas median is the middle value of the data when arranged in ascending or descending order. So, and it is not affected by very large numbers. So median can be used. Mode is the most frequently occurring value. Here all the values are occurring only once. So mode cannot be used. Standard deviation, it is not a measure of central tendency. So that's it for part B questions. Thank you for watching my channel. Please do like, share and subscribe. If you want still previous year question papers of CSIR been answered, you can go to my channel Omni Bio Entrance, and there you will find in the playlist CSIR, where you can find the previous year question papers of CSIR been answered. Thank you.